Hi, everyone, and welcome to the Genetics Podcast. I'm really excited to have not one, but two guests with me today. One is Chris Wigley, the CEO of Genomics England, as well as the senior responsible owner for the NHSX COVID data cell, which is responsible for providing timely and accurate data to the UK government during the coronavirus crisis. The second guest is Dr. Richard Scott, who's the clinical lead for rare diseases at Genomics England, and he's also a consultant clinical geneticist at Great Ormond Street. So we're going to try to cover a lot of ground today because Genomics England in particular has played a central role in the rare disease whole genome sequencing strategy in the UK for a number of years. And now they're starting to play a very central role in a recently announced large-scale COVID-19 human genetics project. So Chris and Richard, I'm very excited to have you, and thanks so much for taking the time to join me today.
Pleasure. Great to be here.
It's great to be here.
Great. So I'd like to start, if we could, by discussing Genomics England's role in leading the NHS into a new era of genomic medicine. So, Richard, I was wondering if, given that you've spent much of your professional career working with patients with rare diseases, if you could just describe what has changed in the past 10 years or so, especially as a result of next-generation sequencing.
It's extraordinary, actually. It's just changed so completely my day-to-day job. So, specifically, if you think what I was doing 10 years ago, I would have been sat in clinic. I knew what next generation sequencing was, and I knew some really cool out there researchers were beginning to pilot its use. And to be honest, it's why I decided to take the sort of career I did. I knew it was coming, but I couldn't access it. So I used to have to sit there in clinic and try to do, if you like, old-fashioned medicine, where you think, okay, let's ask all the questions, let's work out what's wrong with this person, and let's try to be really clever. Let's like be like House and come up with the one really rare diagnosis that no one's come up with, and target the testing. And what was really hard, you think specifically, for example, children with epilepsy. I see a lot of children with epilepsy and maybe more complex problems, and the question is, what's wrong with them? And I probably knew of 5, 10, maybe 20 genes that might be the cause. And I'd have to guess one gene at a time to try and help those families find the answers that they needed. I couldn't even access all of those tests. And then you fast forward 10 years and the tech's moved on, the science, but also the way that NHS England have implemented next generation sequencing in real healthcare. Suddenly now knowledge has exploded. So there are literally hundreds more genes that we know about that could be the cause of these conditions. And now that we can access them through next generation sequencing, and from later this year, NHS England's Genomic Medicine Service using our whole genome platform will be there and in front of me and able to answer these questions. Completely turns things on its head.
Yeah, well, when I was doing my PhD, I worked on severe developmental disorders. I worked as part of a research project called Deciphering Developmental Disorders, and when I was working on non-coding variation, and I worked a little bit on the 100,000 Genomes project at the start. But the questions when I was doing my PhD was always about at what point will it become cost-effective to do this for every child with a rare disease, and it seems like the time, at least in the UK, is now. So, Chris, I was wondering if you could expound on that a little bit. You came into the role of CEO at Genomics England as a relatively fresh pair of eyes because you have actually more of a data analytics background. So you're previously at Quantum Black, which I believe is a part of McKinsey. As the UK for the next decade starts to adopt, as Richard alluded to, a genomic medicine service where I believe the aspiration is that every child, certainly to start with, and hopefully every person with an undiagnosed genetic condition will get access to whole genome sequencing. What are you expecting to see and lead as part of Genomics England over the next 10 years going forward?
It's a very big question, and I think we can expect to see as much change in the next 10 years as we've seen in the last 10 years, if not more, given the shape of exponential curves and so on. And I was really excited to join Genomics England in October last year because it's a really exciting time for us. We've sequenced 100,000 genomes. In fact, because these things get momentum, we've now sequenced about 125,000 genomes. We've passed the results of those back to the NHS, and the NHS Genomic Medicine Centres are pretty much there with getting the primary results of that back to the participants, which is huge in terms of new diagnoses, new insights into those people's lives. And so we're now looking to the next chapter. You know, the first chapter of GEL has been really about how do we get to that finish line. How do we get to the 100,000 Genomes project? And what we're now looking at is how do we scale beyond that? And we think about that in two arenas, really. Firstly, in the clinical arena, and that's the genomic medicine service that Richard's been talking about. But also the research agenda is a huge part of this as well, and those effectively form two big services. So there's a service on the healthcare side, which is patient, doctor, sample, sequence, interpretation, and the return of results. But we can also extract all of the data from that service, critically de-identify it, aggregate it, make it available to researchers in the research environment, and they can continue working on making new discoveries and pushing this field forward. And then critically, we can feed those results back into healthcare. So we end up with what we kind of call the infinity loop, this sort of clinical circle where we're feeding data into research, and then this research circle where we're feeding insights back into healthcare. And really scaling that is what the next chapter of GEL is all about, and having impact on patients, making the benefits of genomic medicine available to everyone. And that's a data and analytics challenge, really, I think, which I think is hence the relationship with Quantum Black and so on. We're already tipping 40 petabytes of data today. That's only going to get more, particularly as we start experimenting with things like long reads, which are about 5 times the size of a short read genome. And equally, it's about algorithms. If we think about what the bioinformatics teams are doing, they're doing data science, and they're trying to, if we think about Richard's every day, they're trying to automate a lot of the heavy lifting for the clinical geneticist out of the process, so that the clinical geneticist can focus on the most important insights and what those mean for the patient, rather than doing all of that heavy lifting. And machine learning and AI are really going to help us do that ever better as we look forward. So yeah, wherever you see cost curves coming down in the way that you've talked about, you see extraordinary things happening. And I think we're seeing that in genetics at the moment.
Yeah, that's great. You mentioned a couple new technologies, machine learning, artificial intelligence, also long read sequencing. What other kinds, on the research side of your infinity loop, what other sorts of things are you all interested in exploring? RNA sequencing, epigenetics, or are you looking at new applications beyond rare disease and cancer?
Yeah, absolutely. I mean, we're very heavily involved in the COVID response, which is, of course, infectious disease rather than rare disease or cancer. Yeah. I mean, we sort of have a joke that the name Genomics England is actually not a great name for what we do, because there's a problem with both of the words, that we're not just focused on England and we're not just focused on genomics. You've touched on some of the kind of multi-omics elements there around functional genomics, proteomics, metabolomics, and there's in fact some really interesting research has just come out of the researchers working in the research environment recently about colon and bowel cancer, and metabolomics playing into that. So we're working with our partners both in academia and industry on pushing the boundaries on all of these fronts, and that's really coming into sharp focus in this program that we have around trying to understand the genomic architecture of COVID-19 in order to get to biomarkers that could help us to stratify different parts of the population for different risk profiles, start to identify biomarkers for vaccine and therapeutic development, and start to lay down some of the infrastructure so that we're ready for future pandemics as well.
Great. I mean, maybe we could jump right into that because it was only announced a couple days ago, the scale that you all are planning. I believe it's 20,000 people with severe responses or moderate to severe, and then another 15,000 who've had mild COVID. If you kind of focus on the high-level goals of this, what's most important to you all? Is it understanding individual response? Is it new drug development? Is it all of the above? It would be great to hear, in 3 months' time, 6 months' time, however long it may be, what you're expecting to see from this.
Yeah, well, I'll maybe talk about the high-level goals, and then I'll let Richard talk a bit about how we're getting after that. So you've touched on one of the key points of the design, which is this stratified cohort. So severe, very mild, ideally even asymptomatic, and then general population. And the more we can start to understand the markers that differentiate those parts of the stratified cohort, then the more powerful it will be. And it's really to get back to those questions of the immediate application in terms of risk profiling, then therapeutic and drug discovery, and then sort of infrastructure preparation. But Richard, maybe you could say a few words about sort of how we're doing that, the different channels, and the where we hope to get to.
Yeah, so we've partnered with a consortium called Genomics, who are experts in critical care and what makes particular people get sick. And it's a question that's not new to COVID. It's been flu, but also other severe sort of insults to the body. They're questioned about why certain people respond in certain ways. And there's good reason and there's precedent to suggest that some of that is genomic. So we've partnered with them, and already they have a network of, now it's 180 or so intensive care units across the country who are recruiting. We've already recruited 2,200 participants who've got severe disease, who've ended up on intensive care. And alongside them, as Chris says, we're going to look at control populations. So amongst the 100,000 Genomes Project, we've got 80,000 participants' genomes that we can look at. They're constitutional, they're germline genomes that we can look at as control groups who are diverse in terms of their demography, including ethnicity, which is going to be really important for us because we know from what's coming out that there's multiple different influences, and what we need to do to make sure the science works and we address those questions is to make sure that our different groups of participants are representative of the patterns of disease that we can see on the ground, and we can see that there is a disproportionate representation of people from minority ethnic backgrounds. And we're also going to be recruiting 15,000 people, as Chris said, with very mild disease or in fact asymptomatic. And those will be people who might self-identify, so in fact people can register their interest in the study on our website already. We'll also, as the antibody programs take shape and scale up, we'll be looking to identify people who've got a positive antibody response. So they've had an infection with the virus, but have been very mildly affected. And again, in that group, it will be really crucial that we match everything that we can about the people who have severe disease so that we've got a really fair comparison and that we're inclusive in who we're looking at.
Yeah, I was actually going to ask about the antibodies because I would imagine that that's played a slightly challenging role in how you design the study because testing has been spotty different parts of the country. I suspect for the severe cases, you're probably not too worried about it because more people are being tested. Are you planning to do at-home antibody testing or working through clinical networks, or is that part kind of still to be determined depending how the different piece of infrastructure play out?
Our plan is broadly to hook into programs that are developing and will develop at really considerable scale rather than us ourselves doing an antibody program. And we're talking to a number of groups who are setting up sizable programs and sort of working alongside them. And there's some mutual gain. Obviously, the prime question, the first question we've talked about is why do certain people develop really severe disease? But also tied to that is why do some people develop an antibody response that peters out after 2 months, 3 months? That's not right. the question that's on the front of the papers today, but it is a crucial question for us all to get out of this and to understand how we can perhaps develop vaccines that stimulate a really good and prolonged response. Understanding the genomic influences of that will be crucial.
Yeah. And Richard said a really important word, though, which is why. And it feels like it's the right time in the curve here to be asking that question. And a lot of the very rapid response work has been effectively looking at correlations. So there are correlations that Richard has mentioned about Black, Asian and minority ethnic groups around, obviously, the age, different age cohorts. There's more controversial ones like smokers versus non-smokers and so on. And we can start to see some of these correlations. But the big question is why? Why are these things happening? And so the intent of the study really is to try and unpick some of the causal factors that lead to these correlations that we're seeing so that we can then plan a much more targeted response to try and address those causal factors, as well as getting sharper insights to be able to kind of use those insights to help society live in a marathon of dealing with this disease, because we can't all stay on furlough and in lockdown forever. We need to get into a process of managing risk over the next 6, 12, 18 months rather than purely avoiding risk.
Yeah, I was actually going to ask kind of what time frame it is that you all are thinking on, because I can see a very near-term potential benefit, which is if you can identify genetic markers of high risk, then you could potentially counsel people that carry those genetic markers to stay home, continue to isolate. But there's also the long-term goal of, my understanding is we've never, as a world, developed a coronavirus vaccine, right? So this is a moonshot in one of the truest senses of the world. So you're also thinking towards the long term of how do we build the scaffold of data that's required to develop something that's never been developed before.
Yeah, exactly. And I think I keep using this analogy, which Patrick, you're definitely too young to remember, but like in the late 90s, when people had like 9.6K modems, it would take ages to download an image, and initially you would get the sort of the blurry image, and then the slightly sharper image, and then eventually like the crystal sharp image. And I think that's very much the approach that we're taking with this program, is that we need to be recruiting as fast as we can. Richard mentioned we've already got over 2,000 people through the very severe channel. We've had over 1,000 volunteers in a few days through the mild channel. So we're really pushing ahead with that as fast as we can. In an ideal world, you would start to derive some insights from those sort of early parts of the cohort. But then we need to keep refining them and making sure that the cohort overall is appropriately powered to actually get to robust insights as that picture comes into focus.
One thing that I've been keeping up to date a lot with because of my interest in rare disease and pediatric disease is this phenomenon of Kawasaki syndrome-like characteristics. Maybe, Richard, I imagine you probably have been following this as well. So there's an observation that children are developing a rare condition or symptoms similar to a rare condition called Kawasaki syndrome at a higher rate in high COVID areas. And I imagine, given you all's central role in rare disease diagnostics, this might also be an area of interest. Is that something you've thought about or considered as well?
Yes, it is. And so we're looking broadly across everyone who gets severe disease, and those children who are getting the Kawasaki-like illness are part of that. And in fact, we'll be, I guess, looking at two levels. First of all, a lot of our learning will come almost at the cohort level, where you see big differences. And for any one individual in the cohort, there won't be that there's a particular answer to why they got it. It will be that they were at greater risk. It may be, particularly among children, that there are certain children who have a rare disease which put them at very high risk and almost out of keeping with the rest of the group. We just don't know whether that's the case yet for the group of children getting Kawasaki disease. It's a condition that lots of work has been done over the years on the genetics, and at the moment we're still really scratching at the surface. It may be that these children are at the sort of the young end of the same spectrum that we're seeing in adults, in truth. But we can only know by looking. And so we'll be taking both the sort of cohort level approach, but also zooming in on those kids and looking in with a fine-tooth comb, if you like, at their individual genomes. And in fact, those are their parents alongside them, to see whether we can see individual signals there.
Yeah, actually, you mentioning the parents jogged my memory that the UK Biobank has recently announced that they're going to be enrolling children and grandchildren even of the members of the UK Biobank as part of a COVID project. Are you all looking at family member-based studies? Or are you collaborating with the UK Biobank on these in any way?
We're looking predominantly at the sort of cohort level. We're looking in these very young participants. We are looking to also recruit their family members, and then we're also in active discussion with UK Biobank about how our cohorts can sit best alongside each other to inform each other's analyses. And this is very much a team sport. This is not going to be solved by one cohort in one country. We're looking for answers that should be relevant globally.
Yeah, absolutely. One thing that I did want to touch on as well that's COVID related as well as Genomics England is the relationship that you all have with the participants in the Genomics England or 100,000 Genomes project. I think it's quite unique amongst especially large-scale research projects. I mean, the fact that you have a 15,000 person goal that's a direct public outreach, I think speaks towards this. I think I've heard you, Chris, say that participants are the North Star. I was wondering if you could maybe in closing just kind of talk us through how that underpins the approach at Genomics England, both in the rare disease and cancer that you focus on traditionally and then in the COVID work you're doing going forward.
Yeah, absolutely. I think it's one of the real strengths that Genomics England has built over the last few years, and it's fantastic to have that kind of muscle, so to speak, in great shape as we go into a kind of crisis situation like COVID. So we've been working closely with the participants over the last few sort of weeks and months to work through some of the impacts of COVID and some of the opportunities for further research. I think the sort of founding point is their data and it's their opportunity. So as I think you probably would have heard me talk about before, the participants actually sit on our Access Review Committee. They are part of the decision-making about what we're using the data for. And Gillian, who's the chair of the participant panel, is fantastically powerful on this in saying, their voice is saying to us, do more and do it faster and while doing it safely and doing it appropriately. But what they're absolutely not saying is, take our data and keep it in a sort of dark storeroom where no one gets to look at it. What they're saying is use it because either I or my child or my family member needs a cure. And the only way that we can get that cure is to get that sort of clinical return on data that we get through collaboration with academia, collaboration with industry, within these kind of channels, these guidelines. The COVID crisis has given us both a need and an opportunity to update a lot of our infrastructure, for example. And we've been working very closely with the participant panel about how we do that, which will be great news for our research collaborators. And what we've also been able to do is link the COVID status data that's reported by Public Health England with the genomic data of the 100,000 participants, so that we will be including the 100,000 participants as an element of this COVID response work as well, working very much hand in glove with them. I don't know, Rich, if you want to add any more specifics on that.
I think that's right. And even one of the things that's really useful for us is having participants who are really engaged in our program, and we can literally have a dialogue about what we're thinking, sense check things. And that's really useful. And to be challenged on, it's interesting, the challenges most often come from why are you being cautious in this way? Well, there's a tendency for, I think, medics and scientists often to be a bit maternalistic or paternalistic about things and saying, no, we ought to be cautious. And the sort of Chris's sort of lock the data in a dark room metaphor, there's a real danger of that. Being challenged on being progressive is really healthy, and it's a great thing to have that link to the participants.
Just a final word on the participants is that particularly as we get running with the Genomic Medicine Service in kind of live operation, we're also working with the participants in partnership with NHS England and Improvement on what that next chapter looks like for patient and participant engagement, because the 100,000 was a thing, whereas the Genomic Medicine Service and the associated research projects that will come up through that, we need to make sure that we're continuing to sort of build on and use this muscle kind of going forward as well.
Absolutely. I mean, it seems like there's a real culture of trust and mutual respect that it's built on top of, right? That the participants are equal members of the Genomics England strategy, and that's a very powerful force because it allows you to bring both the scientific aspect but also the lived experience that the participants have.
Yeah, for sure.
So I'm just conscious of time here. I know you all have a battle against COVID to fight. So maybe just in closing, I wanted to give you both a chance to just share if there's anything you're looking for, whether it's partners, critical roles you're hiring for, or even just your website or Twitter if people want to follow you to keep track of your work.
Well, the big one is volunteers. Richard, tell people how they can volunteer and who we're looking for.
So if people go to our website, you can just Google Genomics England or Genomics England COVID. You'll find our website. You'll be able to read more about the programme. People can also register their interest. There's a big button to click on just to submit your interest. And then more information will follow before people formally sign up to the research programme. We'd love to have people contact us that way.
Great. we'll share that out with the podcast. Thank you both very much. I really appreciate it. Thanks for all the hard work you're doing. I'm sure you're working at 150% right now if you weren't already doing so before.
Brilliant. Thanks a lot, Patrick.
Okay.
Great conversation.
Great. Talk to you soon.
Bye.
Bye.